Can you see me?
So thanks to everyone for joining us in our second BVDzero Scholarship meeting. I'm happy to have today my colleague Gustavo Sabatini with me. And from the feedbacks you sent to us, that parasitology was one of your interested areas, that you were interested. So I asked Gustavo to join us today and give you some essential information on the parasite resistance. So Gustavo, thank you for joining us today. I'll let you introduce yourself shortly and then we go to the topic.
Okay, thanks, Serkan. So it's let's see, it's a very qualified audience, not that big, but very qualified. I can talk to one by name. I said, nice talking to you, William, Emily, Jesse, and Sam. So I am the Global Technical Manager for ruminant antiparasitics at Boehringer. I'm a vet, and I've been working in Boehringer since 2015. At first, I was in R&D, and I started in Brazil. So I moved to Ingelheim around 2 years ago to join the global team and work with Serkan. Well, I think we can do it in a very informal way. So if you have any question while I am talking, just interrupt and ask at any time. So you don't need to wait until the end of the talk, okay? Okay. I'm still trying to get used to this online presentations, because you never get to realize what is the reaction of the audience, and it's not easy to adjust the tone of the presentation in this environment. So you don't keep any question for you, just to shoot out, okay? Let's make it more interactive as possible. So what we're going to approach today is a talk about parasite resistance. And first, we're going to describe what is resistance, how parasites develop resistance. Then I'm going to walk you through some market trends and parasitology challenges. Then we will discuss why it is important to manage parasite resistance and how to do it. And then we will have a final or closing message. Okay, so anyone has an idea of what is resistance or parasite resistance? Yeah, very motivated audience. I can see. So resistance is when you use a product and it does not work as expected. Basically, this is probably the most or the simplest definition of resistance. There are several ones. This one probably is a more fancy one. So it's a reduction in susceptibility of a parasite to the antiparasitic when used at the recommended concentration and according to all of the recommendations for its use. So basically, if you use the product right, according to the label recommendation, and it does not work as it should be, then we are facing a resistance case. So then I have a question for you. So if you treat a herd and you obtain 90% of efficacy, is this a resistance case? Can you say it is resistance or not?
Well, not necessarily.
Not necessarily.
The drug didn't get dosage was not adequate or the drug didn't get to the parasite?
Yeah, that's a good point. But first, we have to understand that there is no product that has 100% of efficacy. You don't need to have 100% of efficacy to have in the label of a product like a claim for I don't know, homuncus or ticks or anything. So we cannot expect that when treating an animal, this product is going to completely kill all the population of the parasites within the animal. This is not what the product is supposed to do, because for registration purposes, let's say, if I have a product that kills 90% of efficacy, 90% of the parasites, I can have a claim. So this is the first thing that is important. And this is stated in the VICH guideline, so at least for endomintic efficacy. The minimum efficacy that you have to call a product an endomintic is 90%. But as Emily was saying, so, and even though if the efficacy is below 90%, would that be resistance? Not necessarily, right? So it's important to carry on an investigation. That's why we have in the company our PV colleagues, the pharmacovigilance colleagues. So you have to go to visit the farm, and you have to carry on an investigation. You have to ask how the product was used. Hold on a second. Laser pointer. Yeah. So you have to investigate. You have to ask how the product was used. So if it was dosed, the animals were dosed based on the proper weight. So you know that it's mainly when we are talking about cattle. Normally, the farmer, he wouldn't weight all the animals to calculate the proper dose and apply the appropriate dose. And that can lead to an underdose, and then the efficacy of the product is not going to be the best one. So this has to be checked. Or what we recommend is, if you are not going to weight all the animals, pick the heaviest one in the herd and dose every animal based on the highest or the heaviest animal dose. So, and then you also have to check the administration routes and products for subcutaneous injection. All the are indicated for intramuscular injection, and that makes a difference in the efficacy if you don't follow the those indications. Also, another important point is to check how the product is stored. Perhaps for temperate climate regions, this is not that critical, but if you consider a tropical area, and the product has to be stored between, I don't know, 10 and 25 degrees, it's not difficult that within a place or a room, the temperature reaches like over 30 degrees Celsius. So that will compromise the stability of this product. Also, is this product, what sort of product was used? What was the batch number? What was the expiring date? How the efficacy was determined? These are all questions that need to be asked before we reach a conclusion that there is lack of efficacy or there is resistance. And how do parasites develop resistance? I'm just trying to fix this bar in a place that it's not going to disturb me to see the screen. So how does parasite develop resistance? So before even any treatment in a parasite population, there is always a resistant individual, and this individual is resistant due to a mutation, and this mutation has absolutely nothing to do with treatment. It happens naturally, you know, in the reproduction process. Something goes wrong, and then there is a mutation. And since those usually parasites, they are very prolific. So one tick, just one female tick, lays more than 20,000 eggs. So that's a lot, right? So they have a lot of chance to develop a mutation, and then eventually this mutation can confer a advantage, like this individual can become resistant to antiemetics. So what happens when the parasites or the animals are treated? So every treatment is a selection process. So what happens is that the frequency of the resistant gene is going to be increased after every treatment, because the susceptible parasites are going to be killed, but the resistant ones, they are not. And then eventually the frequency of the resistant gene is so high in this parasite population that the farmer can realize the lack of efficacy. So you apply the product, and it's easy to see that the product is not working properly. Okay, so let's go through some market trends and probably the most important challenges when it comes to parasitology. First, what is going to happen, or perhaps what we expect that will happen when it comes to animal protein demand in the future. So according to an FAO report, the demand of beef is going to increase from 2005 to 2050 more than around 65%. So this is really significant. And the demand for milk is going to increase, 62%. And why this is going to happen? Basically because the population of the world is growing. So we are now, we have around 7.5 or 7.8 billion people. And by 2050, there will be in this world around 10 billion people. And these people need to eat. They will need to eat. And it's part of our responsibility to help the producers to provide this, provide food for this growing population. Climate change. So what is, if there was any doubt about climate change, last year, in the middle of last year, 3 papers, 3 articles were published in Nature and Nature Geoscience about climate change. What those authors did, they used more than 700 proxy records based on trees, ice, sediment from all continents, from all the world. And they found out that in the last 2,000 years, there was never a shift or a increase in temperature in more than half of the globe, like it has been happening in the past few decades. So what makes those researchers different, or the striking finding of those researchers, is that now we know that the increasing temperature is happening like in the whole globe, like in all continents, and in the past, it has never been like this. And since the temperature is rising, there will be consequences for us. When I mean us, I mean the parasites, because parasites, they like humidity and warm temperatures. And this influence of climate change in parasite dynamic population has already been captured by several researchers in several continents. In Australia, in New Zealand, it has been foreseen that there will be an increase in the challenge of myiasis, flies, and also fasciola. In Europe, perhaps is the place that we can find more papers related to the impact of climate change is in Europe. And also, what has been detected is that we will have a lot more problems with Osteotagia and also fasciola. So it is happening already, right? There's the increasing temperature is going to impact the way we treat the animals, or perhaps the frequency we treat the animals. Some researchers, they foresee an increase in the length of the grazing season. So the animals are going to spend more time on pasture. Consequently, they will be that probably what is going to happen is that they're going to get more than one treatment per year. And every time you increase frequency of treatment, you increase the selection pressure for resistance, and this is no good. New products development. So, from the ideation until the product is available for the farmer to treat the animals, it takes around 10 to 15 years. It's a super long process, and it is necessary a cross-functional team to work on this. So first you have to find or discover an active pharmaceutical ingredient, what we call API. Then you have to formulate this API in a stable formulation, which is also another challenge. Then you have to go through all the studies to prove efficacy, safety, not only for the animals, but also for humans, if we are talking about production animals. And then you can, put all this information together in a document that is called a dossier, and you submit this to a regulatory authority. And after a couple of years, normally 2 or 3 years, if everything is okay, the regulatory authority is going to give you a green light to commercialize this, your new product. So the cost for the development a new product, we are talking here is about a product based on new APIs, new mode of action APIs. The cost for this development is around 35 to 40 million euros. It's a lot of money. So, and what is happening now is that the regulation, mainly the regulation related to food safety and ecotoxicity, are getting more stringent. What does it mean? What is the consequence for the development process is that the development is going to get longer, and it's going to be more expensive, because companies will have to do a lot more studies to prove the food safety and that the product is not going to harm the environment. So I want to draw your attention to this graphic, and just pay attention in the X bar here. So here you have year of introduction, and in the middle of the graph you're going to see different classes of antiparasiticides. So the first endectocide, which was ivermectin, the first macrocyclic lactone, was launched in the beginning of the 80s. And still now, until now, there was no launch of a new mode of action endectocide. So we are using this products from the same class since 1980s. And you know what happens? Ivermectin, abamectin, doramectin, eprinomectin, moxidectin, they all have the same mode of action. With what it means for resistance matters is that if there is resistance to doramectin, there will be resistance for ivermectin, abamectin, and any other compound of this chemical group. So we have been trying to develop a new mode of action product since the early 80s. It has been like 40 years, and we haven't succeeded yet. So that's important information, because we need to safeguard the efficacy of the products that we have available nowadays. We cannot expect that a new product with a new mode of action is going to be developed and launched every time there is a resistance issue in the field. This is not going to happen. Parasite resistance situation. See, resistance was first detected in small ruminants, and some would thought that it would be restricted to small ruminants forever. But nature is very powerful, and what happened is that resistance appeared also in cattle, and nowadays we have it's also very common in horses, in equine, and it's beginning to become more important in companion animals, because there is no other way. Historically, we know that if you treat against parasites, eventually they will find a way to survive. We cannot, it's absolutely impossible to prevent completely the development of resistance. What we can do is we can delay the resistance development, but we cannot prevent it. So nowadays, probably you are aware, we have there are resistant parasites in small ruminants, in cattle, ectoparasites that are resistant to not only one API, but mold-resistant ticks, mold-resistant flies, mold-resistant gastrointestinal nematodes. The situation is, let's say, not ideal, to say the least. So let's just make a pause here and recap. So first, we talked about an increase in the demand for beef and milk, so the animal protein. If we are going to need more milk and more beef, consequently, what the farmers are going to do, they're going to intensify their production systems, right? So they're going to put more animals per square or per hectare. And when they do this, every time that this happens, the animals, they are going to eat closer to the dung patch. And by doing this, the chance for them to get infected is going to be higher. So consequently, they will need more antiparasitic treatments. What is going to happen? Resistance situation is going to be worst, right? Then we talked about climate change. So we saw that what has been foreseen for researchers is that the parasitological challenge is going to be increased in the future. Consequently, the animals will be needed to treat it more frequently. And that's going to make the resistance situation even worse. And to how another information, we cannot develop new products as fast as the parasites develop resistance. So what I told you is that we cannot expect that a new product with a new mode of action is launched to control a resistant parasite every time there is a resistance case in the field. And resistance is already a super big issue. It's probably the most serious challenge that we have in parasitology. So how this system, everything that I told you until now, works for you? Can you be optimistic about the future?
Is it a general question, Gustavo?
Yes, a general question. I
mean, or about the parasite control.
I mean, yeah, of course, I'm talking about parasites here.
Yeah, maybe I would like to say welcome to a few colleagues because they had just joined. Maybe they would miss the introduction of your call. So maybe Emil would say something on this. And then...
Well, I'm optimistic because I know there will be an answer coming in the presentation because there are ways already applied to. It's not as easy as giving a drug to an animal, but it's a lot of information, and I like it. It's population medicine, so it's coming.
Yeah.
Hopefully, we'll have the answer.
Well, this is the point. Thanks. So
it's Emil. I removed the E's to help you.
Yes, sir. Yeah, I thought it was a strange Emily. But okay.
Emily is for female.
Yeah. So Emil, that's exactly the point. I mean, the thing is that we need to control parasites in a different way from now on. Because if we don't start doing things differently, then there's not going to be a solution. So if we continue to continue to control parasites based only or exclusively on anthelmintics, the situation is going to be unbearable. And so, but you're right. We need to do things different. We need to manage parasite resistance. First, because we need to keep the efficacy of the products that we have currently on the market. We need to keep the efficacy of those products, high enough or for them to be properly used by the farmers. This is a strategic issue for us, because nowadays, there's not a single farmer that will think about the biology or epidemiology or the climate to create or elaborate a parasite control program. It's very difficult. What normally is happening in the field is that let's use anthelmintics and that's it. That's the approach nowadays. And and things have to be different. Otherwise, it's going to be unfeasible from a financial perspective to graze animals. So we know that either if you don't keep the efficacy of the products that are available now, either we will have in the future no efficient product, or if there will be a launch or a new development, which is very unlike, because I showed you historically, it's not easy to, historically, there's not a new launch in the last 40 years. So it's a long time. But even if there is a new development that is about to be launched, it's going to be very expensive, right? So it's important for the farmer to manage resistance. If the farmer wants to keep his business feasible, he has to think about it. There is a social responsibility element here as well, because as we talked about it a couple of slides ago, so there is the population of the world is going to be around 10 billion in 2050, and those people will need food. And for the producer, it's important not only because from a financial perspective, he's going to be able, if he manages resistance, he will optimize his productivity and will also minimize potential environmental impact. Because when you manage parasite resistance, you tend to use less chemicals. You tend to treat less frequently the animals. And how do we manage resistance? So we have to implement best practices. And how do we do this? So first, in general way, it's important to identify the parasite in this specific farm. So there's one thing important here. There's not one size fits all solution. Things from now on are going to be different from what was in the past. When the macrocyclic lactones were launched, it was a revolution. When Ivomec was launched, it was a revolution. Because until the beginning of the 80s, to control parasites it was not easy. So why it was not easy? Because there was not a product that with one single shot would kill all internal and external parasites for a long period of time. And when Ivomec was launched, it was like, oh my God, this is incredible. But because parasite control became so easy, I think everyone got used to it and started to overuse anthelmintics. And as a consequence, we are facing resistance issues nowadays. So now we have to take one step back, and we have to go back to the basics of parasitology. So now to elaborate a proper parasite control program, you have to understand, you have to identify what is the parasite that is causing the main problem in your farm, in this specific farm. Then it's important to check what is the resistance profile of this parasite population. And there are several ways to do it. If it is, if we are talking about gastrointestinal nematodes, you conduct what is called a oh my God, I forgot the name of it, fecal egg count reduction test. So how is this? Before treating the animals, you separate two groups of animals, or 3 or 4, or as much products you want to try. Groups of 10 animals. And then just before treatment, you collect feces samples. Then you run a EPG, just a McMaster, to check how is the to get an idea of the parasite burden of this group of animals. Then you apply one product to each of those groups. Preferably or ideally, you're going to use one product of different chemical class to each of those groups. And then you're going to, after 15 days, you collect feces samples again, and you do McMaster again. So then you're going to find out which is the product that is working better in this property. Then you will find you have the answer for what is the right product to be used. Then there is a concept that is called refugia. I don't know how familiar you guys are with refugia. Have you heard of it? Some of you yes, some no. So basically, refugia is to leave a couple of a small percentage of the animals in the herd untreated. There is the next slide is we can talk about it in the next slide. I will tell you the importance of refugia. So by leaving a small proportion of the animals untreated, you're going to create a refugia for the parasite population. What it means, there will be susceptible parasites in the environment, and then the resistant genes from the resistant parasites will be diluted. So that will make the resistance development process to be delayed. Then you have to consider what is the best time for you to treat the animals. Thinking strategically, you need to understand, and this varies from region to region, from farm to farm. You need to understand when the parasite population is going to increase. And then just before that, you treat the animals. So what we want here is to avoid that the environment gets super full of infected stages of the parasites. Then you have to pay attention to the product administration, like follow the label. So weight the animals, or at least those based on the heaviest animals. And last but not least, it's if possible, you have to implement some pasture management initiatives. A couple of years ago, it used to be recommended to dose the animals and move to a clean pasture. When you do this, you're going to take the animals to a clean pasture, and this clean pasture or this clean paddock is going to have only resistant eggs, which is no good. So you want the eggs to be in a paddock that there is also susceptible eggs. So then the resistant genes are going to be diluted. So what has been promoted by, I don't know, William, if you have heard of scopes. So this is that's right. So this is exactly what has to be done. So you have to use the right product with the right dose in the right animals in the right time. And I just added pasture management to the scopes mode here.
Gustavo, I have a question in the chat box from Jesse.
I
don't know if you can see that.
I can't. Yeah, thanks for.
I let you read it, or Jesse, you can just unmute yourself and.
Yes. You were typing refugia leave circa 20% of the animals untreated. So is that in order to reduce upcoming resistance against the product you are using? Or why would you leave 20% of the animals untreated?
Okay. So perhaps I can explain to you with the help of this slide. So if you imagine this is a parasite population in a herd, okay?
Mm-hmm.
So in the parasite population, there are susceptible parasites and resistant parasites. So if you treat those animals, like 100% of those animals, the susceptible parasites, they will be killed by the treatment, but the resistant ones, they will survive. So this is what you're going to get in the pasture: eggs from super resistant helminths or parasites, let's say. Okay.
Yes.
In the next, let's say, grazing cycle, the animals or the cattle, they're going to be infected or infested, because they are helminths. They're going to be infested with resistant helminths only, and it's going to be very difficult to treat these animals to make them free of helminths. Okay.
Yes.
So, and if you leave, so consider now that instead of, it's the same population, okay, but instead of treating 100% of the animals, you leave 20% of them untreated. So let's say you're not going to treat this, and this. So what is going to happen is those the resistant parasites are going to be here,
Mm-hmm.
okay, they are going to survive, but in the pasture or on the pasture, there will also be eggs from susceptible helminths, and this is going to dilute the genes, the resistant genes in this population. So that's why. So in the next grazing cycle, there will be animals that will be infected with susceptible parasites and resistant parasites. But the frequency of resistant genes is going to be, let's say, controlled. Is it clear?
Yes. Yeah, it's clear. So it's in order to reduce the development of resistance in the herd.
That's right. That's
Okay.
right.
Thank
So
you.
what happens is that this has been studied a lot, and there are a lot of publications in the small ruminants. So it is known that it works in small ruminants, and it's also cost effective for the producer. Is very good because he is going to spend more money buying anthelmintics. He's going to use less products, and also for the environment is super good. And we know that it has been proved that this concept of refugia slows the development of resistance. And by leaving 20% of the animals untreated, you don't compromise the productivity of the herd. Papers usually the papers about refusion in small ruminants, they treated around 25-30% of the animals. So 70-65% of the animals are left untreated, and still the productivity is not compromised. So there's super interesting results. But I have to be honest with you. There's not much information about refugia in cattle, but only because it's super expensive to do studies with cattle. They are bigger and they have to eat more, and it's just more expensive. But we have to do something. As I told you, if we don't start doing things different in cattle, in a few years, it's going to be impossible to graze animals. I mean, as it has happened, it is already happening in small ruminants. There are producers that they just close the business because there's no way to treat the animals against parasites. There's nothing to do. They have a multi-resistant strain in their farms, and they don't know what to do because there's no product that's going to work. So we have to prevent this situation to happen in the cattle realm. Sure, just aware of the time, Serkan. I will just say a few words to close the session. Sure, but Emil, I'm with you. I'm optimistic, but I think it's important that every stakeholder in our business, in this in the ruminant segment, plays a specific role. If every stakeholder plays its role accordingly, I think we're going to succeed in the fight against parasites. For example, the animal health industry. I think it's important that the animal health industry keeps investing in new products development, perhaps in products that are green and you know, with no residue, with no impact in the environment, and also we need to promote best practices for parasite control. We need to help the farmers, or help to instruct the farmers to get away from this old-fashioned way to control parasites that is which is so-called the solely or exclusively based on chemical solutions. The scientific community, it's important that the scientific community develop new techniques or strategies to keep the efficacy of the products that are available in the market now. There are examples of this. I mean, have you heard of Famasha for sheep? And no, Famasha is super interesting. There was this researcher in South Africa, Jan van Wyk, and you know that for sheep, the bad guy is the Emoncus, and Emoncus is the bad guy, and the Emoncus is the bad guy because he's a vampire. It's a blood-sucking parasite, and it really damages the physiology of the host.
Gustavo, can you write in the chat box, just for me to curiosity, the name of it? The
name of the this technique.
Yeah.
Yeah. As soon as I can find the chat box, I can do it.
Okay.
Where is it?
If you go down.
If I go. Yeah, but the thing is, I have a different screen because I'm presenting. I don't have access to the chat. I can get out. Okay.
You can go up in the or down. Sam already. Okay, thanks, Sam. Sam already wrote it. Okay. Okay.
All right. Okay.
Great. Okay. Just want to make sure that everybody's.
Yeah. And what happens is when the sheep are infected with Emoncus, they present anemia. And this guy in South Africa, he established a relationship between anemia and the need of treatment. So he has a card. You can see me because it's so the sun is shining, and but he developed a card with several grades of anemia, from the super red, the normal mucosis on TDI, and then 5 degrees of shading in red. If the animal if the eye of the animal is not as red as it's supposed to be, then this animal should be treated. But if the animal is okay, you know, there is no anemia, this animal should not be treated. So this has been a super achievement, a great finding, and it works. It works very well. So then you can identify the animals that need to be treated. So it's a targeted, selective treatment. There's no, we cannot do this in cattle. It's just not feasible. It's not, you know, you cannot just grab the head of a cattle or of a bovine and check for the eye mucosa. It's just not feasible. And the most important parasite or ailment for cattle is Osteotagia. That it's not exactly a blood-sucking parasite. So it does not work very well in cattle, but it works very well in sheep. So we need something like this to cattle, and the scientific community can help developing, you know, those techniques or strategies to keep the efficacy of the products high. And the producers, they need to understand that nowadays it will be necessary a little bit more than just apply antiparasitics to the animals. So, you know, it will be needed to understand biology, epidemiology, strategic treatments related to the parasite dynamic population, pasture management when it's necessary. We need to make them understand that. And if everyone does its homework, then we will be successful. We will find or reach sustainability in ruminant parasite control. I think this was the last slide. Yes. So thank you very much.
Thank you, Gustavo. Now I ask everyone to turn on the cameras. Time for questions and some catch up. Yeah.
I can close this window because it's, would you give me like 2 seconds, or it's good like this?
We are not cold, but if you can. Yeah. Oh, I see Bruna as well from Brazil. Gustavo, we have a colleague from Brazil.
Oh, Bruna, tudo bem?
Oi! Cheguei meio atrasado.
Que coisa, hein?
Okay, so Sam is leaving. I'm just saying goodbye to him. So any questions to Gustavo or any comments? Was it useful for you guys?
I've
what
got
do
a
you think?
Instead of trying to produce new anthelmintics, is there any effort within the industry to produce animals and livestock which are genetically resistant to parasites instead?
Yes, there has been some effort in this direction. Usually Bos indicus are more resistant to parasites than Bos taurus, and there is already a difference. But the problem is that every time you select an animal for parasite resistance, it's like the cattle is going to be resistant to parasite, and so it's important to clarify. Every time you identify those animals and you try to select them, this characteristic is just selected in the opposite of productivity. Did I make myself clear? Every time you select for parasite resistant, you suffer because the animal is not going to be as productive as the susceptible bovine. But there are exceptions for that. There is in Australia